All right, thank you again to our presenters for their excellent presentations, and we will now have our final question and answer panel to include all our presenters, and also joining the panel we have Dr. Nilufer Tampal, Associate Director for Scientific Quality with the Office of Bioequivalence in the Office of Generic Drugs, as well as as well as Rachel Erdman, JD, Regulatory Counsel with the Division of Policy Development with the Office of Generic Drug Policy within the Office of Generic Drugs as well. All right, and as before, attendees in the room, you are invited up to line up at the mics to ask any questions. And for those online, if you haven't had a chance to submit your questions using the on-screen link and QR code, please do so now, and we'll answer as many questions as time allows. All right. So again, in the room, this is the last chance to ask the experts while you have them here. So at any point, please feel free to come up. And in the time being, we'll go ahead and start with our online questions then.
All right, so our first question is for Dr. Greg Huang. So for lyophilized product, if RLD composition doesn't provide salt form quantity, but only includes free base quantity per vial, as an ANDA holder, do we have to specify the salt form quantity in composition?
Yes, both the salt form and the free base concentration and the name should be included in the composition table. And also it should be clarified in the labeling.
Thank you for that response. And now we have a question in room.
So the question is for Dr. Huang as well. So is excipient function part of the Q1, Q2 evaluation? So for example, if we match in all the other chemical characteristics as well as the quantity, however, we list the different primary function as the RLD holder, would that make us not Q1 equivalent?
This is an assessment question. Basically, the terminology may be some different in the naming, but it should not affect the Q1, Q2. Q1, Q2 is based on the qualitative and quantitative information, but we understand that some different applicants may use slightly different naming, but the role should be equivalent. And
so
also,
for example, like BHT, we list as antioxidant, they list as preservative, but this is an extreme case since they're both exceptional. But in that situation, they're quite different in terms of their functions. Would that be okay?
Yeah, well, you might get a question if we are not clear about your description of the role of the excipient. Yeah, you might get a question in the CMC review.
Okay.
Thank you.
Thank you for that response. We'll go to another online question.
All right, so we have another follow-up question for Dr. Huang. Should all inactive ingredients be included in the outer pack or container label?
Yeah, all the ingredients should be included in the labeling. The name, the list, the full list.
Thank you for clarifying that. We'll go for another online question.
All right, we have another question here for Rachel Erdman. So should applicants round the amounts in the statement of ingredients in the labeling?
Well, thank you. Generally, no, we don't request that you round the numbers. The numbers, significant figures should match what's in your composition statement. Thanks.
Thank you for that response too. We'll go ahead and continue with our online questions.
All right, this question is for Dr. Fang Lu. If OPQ identifies GMP issues related to an API supplier, but the applicant showed that the API batches do not have concerns related to GMP issues, will FDA accept the biostudy?
it's case-by-case scenario. Semi-adjudication we can consider.
Thank you. And we still don't have any takers in the room quite yet. Oh, okay, actually we do. All right. So thank you for coming up, and please go ahead, ask your question.
This was a great program. This is a hypothetical question to anybody in the panel or for the entire thing over here. I've always seen that, okay, brand drugs, and now we are talking about generics. So in case the brand manufacturer decides to make the first generic, does it mean that he has to just do a label swap? Like, does he have to do an ANDA with just a labeling requirement, or what happens there? You know, if it's the brand, like let's say you can take, I don't know if it's Mylan or Teva for the EpiPen, they made the generic epinephrine injection, right? Was there an ANDA process for that, or was it just like any of the products, like let's say any of the monoclonal antibodies or any of these really complex drugs, like do you have to do another ANDA process even though it's the same manufacturer who's got the brand approved?
Okay, so I'll try to.
Kind of, that's why I didn't want to ask anything, but it's the question that's been intriguing me the whole 2 days, you know, like, because there is a commercial value in having a generic product because you don't want to lose the market. So if you decide to, because it's going to expire, it's going to, so I don't know what's the FDA take on that.
Okay, so what you are referring to is really the authorized generics. That's what we call as the authorized generics. So you, as the innovator, when you want to market this as an authorized generic, all you're doing is just changing the labeling in the front. Then there is no ANDA submission. There is not that I'm aware. I wouldn't say there is no ANDA submission for that. But if you are marketing an authorized generic, you have to, I'm not an expert here in this authorized generic field, but in terms of you have to inform or there is a authorized generic site, you get an NDC number and those things. So you may have to just look into the FDA's, some website for unauthorized generics for additional information. Thank you.
All right, what a great example. If anyone else here has some questions they've been wondering about, now's a great time. We'll do another question here, actually, yes?
Hello. Yeah, I have a similar type of question. So, some of these drugs which have been approved long back, ages back, now as you get a new drug or generic drug product out, you go through a full characterization, and there's a whole bunch of specifications, including impurities and stuff like that. And as part of the ICH guidance, with the impurities, you sort of show some of those things. What if the brand themselves have those impurities which are higher than that, the ICH guidance? What happens in those cases? And it was not in the specification because it's approved ages back.
I think maybe this is a little out of scope of this particular session, and I'm not sure if anybody else wants to answer, but I think this is a little out of scope on this ICH and impurities.
Sure. All right. Well, thank you, Sil, for being willing to ask and that response. And actually, we'll move on to an online question first. Yep.
All right, this question is directed towards Dr. Priyanka Ghosh. The first thing they said was, thank you for providing a very informative presentation. Regarding the characterization-based BE approach, emphasis on the IVPT and IVRT aspect for establishing the BE of topical products, will there be a bioequivalence data summary table that is formatted and tailored for this type of submission, as the current model bioequivalence data summary tables seem to gear towards clinical data? Thank you.
So at the moment, we do have some summary tables available for IVPT and IVRT that are accessible through the FDA's website. However, if you run into challenges as you are trying to utilize them, I would encourage you to let us know so we understand where the challenges exist and we can update them so they are easier to use.
Thank you for that response. And we had another in-house question.
Hi, thank you for coming out today. My question is about the ICH M13 and harmonization. So first, we thank the FDA for driving and leading some of that harmonization, all those efforts. We understand the challenges of that. Where I work at AAM, we work with one of our partners, IGBA, the International Generics and Biosimilar Association, and we advocate on certain issues. One of the issues we talk about is single global development. And with that, we think about three pillars. We think about the legal frameworks in different jurisdictions. We think about harmonization at BE, which you talked about. We also think about single global development or single global comparator. On slide 76, you mentioned multiple comparator, multiple regions. Can you give us some thoughts or some ideas of how we move the concept of single global comparator down the line, or how we push that concept a little further so we're not doing multiple comparators in multiple regions for the same product? I hope that's not out of scope.
Okay, I will get a start, and Nilufer can always add on. Yeah, we heard you. We did hear that feedback. But however, in the guidance, we did talk about the selection or acceptance of the comparator product. It is really bound by the regional legal jurisdiction. So it's like ICH will not be able to harmonize that in terms of, which acceptance comparator will be accepted by which regulatory agency. So the solution we provide is really what you mentioned is you could, in a single trial, you can include more than one comparator. And if you only rely on one for so the multiplicity, we also talk about multiplicity adjustment. In that case, you don't really need to adjust the multiplicity if you only plan to submit the set of data to one particular region for the one particular comparator. I don't know if that addressed your question, but that has been discussed, and we just don't have the bandwidth, I think, in ICH to harmonize that. Do you have any?
Yeah, it's more than the bandwidth. I would say that's just a guiding principle of ICH, that we harmonize on the whatever standards, on the scientific and technical standards of whatever the expert working group is working on, while staying within the jurisdictions or the regulatory framework of the different regions. So since every region, like EMA, USA, each one requires a very specific reference standard against which the study needs to be done. That's something which is dictated by the regulations, and so that's why it's out of the scope of ICH. So yeah, it's not ICH, maybe some other way to do it.
Please also consider comparator variability from scientific perspective.
All right, thank you all for that. We'll go to one more in-room question here.
I have a very simple question. Does the MIE pilot program cover only generic drug or new drug as well, or maybe drug device combination as well?
So I will start, and Lei can add. The MIE pilot program currently covers generic drug. You're asking for drug device?
New drug and drug device combination.
So in new drug, there is an MIDD program, which has similar function to discuss the modeling. So for the MIE, it's the one I'm showing, it's mainly focused on generic drug.
Thank you for that clarification. Now we'll do some online questions.
All right, so our next question is for Dr. Lei Zhang. So prior to the M13A, I was instructed by FDA to conduct both fasting and fed studies for my product, which I think is non-high risk based on M13A. Now M13A is under implementation. Do I need to submit a controlled correspondence asking the FDA to adjust the recommendation, or will it be sufficient to rely on the revised guidance and move?
Yeah, thank you for this question. Yeah, now we are in the middle of revising all the product-specific guidance. I would suggest look at the forecast list. If it is being listed as being in the plan for revising. That's a hint about what direction FDA's current tentative thinking is. But you can also wait till the PSG is published. You can follow that. I do not think the person need to submit another control correspondences, but if they choose to, it's okay. But I don't think it's really necessary. They should follow the FDA guidance. Thank you. Updated guidance.
Any more comments from the panel? No? Okay, we'll move on to the next online question.
All right, this question is for Dr. Nilufer Tampal. So with this, what should a firm do if it is about to conduct studies under both fasting and fed studies based on previous PSG recommendations for a non-high-risk product? Should it obtain permission from the FDA to drop the fed study, or should it just do it on its own?
No, if you are planning to do 2 studies and if the revised guidance is already posted, which is recommending maybe 1 study, then that guidance is what you should generally be following. So is that the question? I'm sorry.
I believe so, yes.
Yes, okay. Yes, thank you.
All right. So we'll go ahead and do another online question.
Excellent. This question is directed to Dr. Fang Lu. So if applicant uses alternate API source because of CMC issues with the original API supplier and submits CMC data along with the comparative dissolution data during a pending ANDA, will it suffice requirements for ANDA approval, or does the applicant have to conduct BE studies with alternate API source batches?
Can you repeat this question?
Of course. If an applicant uses alternate API sources because of CMC issues with the original API supplier and submits CMC data along with the comparative dissolution data during a pending ANDA, will it suffice requirements for ANDA approval, or does applicant have to conduct BE study with alternate API source batches?
First, to figure out if the API source or supplier has any CGMP issue, I will refer you ask OPQ.
Yeah, we would suggest that you ask this in an appropriate response to FDA controls correspondence, since this is very application specific. Thank you.
Thank you for clarifying that. All right, and again, we'll continue with our online questions, but still in the meantime, if anyone in-house has anything, feel free to come up. The invitation is still open to come to those mics. So we'll go ahead online.
All right, so this question is for Dr. Greg Huang. So if we have complex inactive in formula, is it okay to list only flavor name, or should we include all components of that flavor?
All components of the flavor and the quantitative information should be provided in module 3P1 composition. Okay, but in the labeling, you may not need to provide that detail about the flavoring agent or coloring agent.
All right. Next question from online.
All right, so this one is for Dr. Yuqing Gong. Regarding MIE meetings, can we in industry use that to discuss modeling certain aspects of device design for a drug-device combination product? We invest a lot of resources into comparing these products, and we could use a modeling approach, including using virtual study subjects for comparative use human factors, for example.
Yes, the answer is yes. You can discuss those type of questions in the MIE.
Thank you for clarifying that. And so we'll keep on going with our online questions.
All right, so this question is for Priyanka. While I understand your presentation was limited to topical products and their respective draft guidances, they, however, list other dosage units that are not within the scope of these guidance documents. When working with IVRT and/or IVPT studies for these other dosage forms, for example, transdermal systems, is it recommended to apply as much as possible within these guidances to the IVPT study design, or are there additional guidances for these dosage forms expected in the future?
Okay. Yes, my presentation was focused on topical products applied to the skin, where we do recommend IVPT and IVRT studies within the scope of characterization-based PE approaches. When it comes to IVPT, and since transdermal dosage forms were mentioned in the question, I think we already do have a guidance, the product quality guidance for transdermal products, that has recommendations related to how IVPT studies can be conducted, for example, to support evaluation of heat effect of a generic transdermal product compared to the reference listed drug. In other situations, I think we currently do not recommend IVPT studies specifically to support product approval. So if you are wanting to conduct such studies, then I would encourage you to chat with us using the pre-NDA pathway.
Thank you for that response. We'll go ahead and go for another online question.
All right, this question is directed towards Rachel Erdman. FDA recommends listing the established name of an inactive ingredient in the statement of ingredients in a drug product's labeling. What if there is no official designated name under Section 508 of the FD&C Act?
So if you're saying that there's no official name and there's nothing in a compendium or anything like that, like USP, then you can generally use the common the common name of the drug, and of course just double check to make sure that that's acceptable. Thanks.
Thank you. Do we still have some more questions coming in online?
I can get some.
Okay.
So we have this next one is directed towards Dr. Fang Lu here. Can we consider the dissolution data from the comparative dissolution testing supporting the API source change as the same as that of in vitro dissolution testing for supporting waiver of in vitro BE study requirements for the lower strengths?
No, it's different. Number one, let me answer. Number one, for waiver of your lower strength, the study should be conducted with. Let me figure out. It's different with T versus R. For the other situation, like for API source change, this should the in vitro dissolution conducted with pre-change with post-change of T. Those different is T versus R, pre-change T, post-change T. That's different.
Thank you for that. All right, and let's see, do we have, okay. Any other last call for questions here in the room? Again, this is the last chance, don't miss the opportunity. All right, well, in that case, we'll go ahead and thank our presenters again and panelists for all their helpful information. We'll give them another round of applause. Okay. And panelists, you can go ahead and take your seats, but everyone else, if you want to stay put just for something special, and that is our closing remarks from Dr. Robert Leinberger, who serves as the Director of the Office of Research and Standards, or ORS, in the Office of Generic Drugs, or OGD. Dr. Leinberger leads OGD's implementation of the GDUFA science and research commitments, including internal research activities and external research grants and collaborations to ensure the therapeutic equivalence of generic drug products. And ORS also provides pre-submission advice on complex generics through pre-ANDA meetings, product-specific guidances, and correspondence responses. So I'll hand it over now to Dr. Leinberger.